Hi, I'm Cheryl, a cell biologist working with Eurofins Discovery and our Translational Biology Center of Excellence. I'd like to ask you, what do pizza boxes, raincoats, and frying pans have in common? They all contain one or more PFAS, a large family of chemically stable structures used in numerous consumer products. Because of their stability, some of these chemicals build up in the air, water, and our bodies, where they eventually pose a health risk. These concerns include development, immune function and immunosuppression, thyroid function, fertility, and others. Did you know that it is possible to screen for and predict human toxicities using cell-based systems? Eurofins Discovery's BioMAP Platform with Toxicity Signature Analysis has been used by the U.S. Environmental Protection Agency to understand the impact of diverse PFAS molecules on human biology.
Hi, I'm Julia and I'm a translational biology specialist at Eurofins Discovery. Did you know that BioMAP systems are unique to Eurofins Discovery and capture tissue-level biology? Cells from healthy human donors are used in mono or complex co-cultures together with a cocktail of growth factors and cytokines to produce these systems that model normal and diseased tissues. Three examples here show systems modeling T cell activation, B cell activation, and macrophage activation. All BioMAP systems are validated with clinically relevant therapeutics. The Diversity Plus BioMAP profile shows biomarker responses across 12 systems capturing a broad range of human biology. Each test agent is evaluated across 4 concentrations, producing a unique fingerprint for comparative analyses. Significant biomarkers are annotated and arrows indicate antiproliferative and cytotoxic effects.
Hi, I'm Frauke and I'm a translational biology specialist at Eurofins Discovery. Did you know that we can predict human toxicity mechanisms in vitro and by this de-risk your molecule for clinical adverse effects? The BioMAP Toxicity Signature Analysis identifies specific patterns of biomarker changes that have been developed by comparing key reference drug profiles with known adverse outcomes and understanding the underlying mechanisms and targets involved. The toxicity signature analysis performed on the different PFAS profiles revealed that PFOA and PFOS had no detectable toxicities, whilst others were positive for the immunosuppression, organ toxicity, and skin sensitization signatures. Get in touch to learn more about this analysis. For more information about BioMAP, our Translational Biology Center of Excellence, or for the complete PFAS case study, visit us online.